In this lesson, I would like to demonstrate how to do a monohybrid cross. In your Biology 1 classes, we will be doing a particular genetics problems. And as we look at this little simple, nice cross can be pretty simple for us. Now, a monohybrid cross is a breeding experiment in which two individuals have one gene in common. So perhaps this is a gene for flower color. So, for example, here we're going to say we have a purple cross with a red. So we have a purple colored flower plant, and we're going to cross it with a red colored flower plant. Now, there's a few things you need to actually know about these individuals. Now, what does it matter for us right now if this individual's purple or if it's red? We need to know its genetics. We need to know what its genotype actually is. So let's, for this cross, assume that purple is dominant, and we actually have a homozygous dominant purple plant. So for this particular instance, and I want to choose a different color here, we'll use blue. We're going to say he is big P, big P. Now, we're going to only cross him. Red, let's say, is recessive. We're going to say it's little p, little p. So in this case, when I go to cross these individuals, the first thing I actually have to figure out is what kinds of gametes that are going to produce. Well, obviously for us, if I use a different color to kind of circle around here, that the purple individual can only produce two gametes. Remember that the gametes that can produce, that each gamete possesses one allele of each gene, and there are two alleles per gene. So we can produce one big P and another big P allele, one little p and another little p allele. So in this case, if I were to show you guys how this works, I can take one individual who's big P, another individual's big P, and cross them to the gametes of the other. So if I were to put these together and allow them to breed, how will this happen? A big P crosses a little p. Remember, big P is purple, and little p is red. This is standard dominant, so what we're actually going to get is an individual who is this, and this one. Now, note that I'm crossing the first one. The big letter goes first, and the little letter goes second in this case. So big P, little p, big P, little p. So in this case, every one of these guys would all be purple. So if we were to sit here and make the genotypic ratios of these individuals, well, the genotypic ratio tells us three things. There is three numbers. The first number is the number of homozygous dominant offspring. If we look, we have a heterozygote, a heterozygote, a heterozygote, and a heterozygote. So there are 0 homozygous dominant individuals. There are 4 heterozygotes, that is the second number, and there are 0 homozygous recessives. So this is a 0-4-0 ratio for genotype. They are genotypically heterozygotes. But a heterozygote looks just like its parents. So when going to make a phenotypic ratio, you want to sum the first two numbers, and that would be 4. And so the phenotypic ratio is all look dominant, none look recessive. So 4-0. So a basic genetics problem like this would follow with these things, and I can do others. So what we're going to do is I'm going to erase these, and I want to give us another more complex example of this, and we'll do another monohybrid cross as we kind of think, how do we set this up? How do we build the cross? What do we do with it? And trying to figure out these guys by information, it can be kind of tricky. So I'm going to start out orally, and we'll work out the problem. So in this case, we have a black Labrador Retriever breeding center that we want to breed 2 Labrador Retrievers. So if you're at home working on this, you want to write down that we have a black lab, and in black labs, black is dominant. We have a homozygous dominant black lab, and we're going to cross this with a chocolate lab. In chocolate labs, what do we have? Well, in chocolate labs, we have chocolate being recessive. So this is actually, it may not seem like flowers, but if we think about it, if chocolate, we'll use purple here, if we have a chocolate lab as one, chocolate, and black as another, well, black is big B, chocolate is little b, little b. Well, look at this. When we got this one again, this is exactly the same as what we did before, and they all look like that, all. And it's the exact same problem. So we can see what we've learned helps us solve a wide variety of problems. But let's change things up a little bit. Imagine now that I have this black lab, and we have all heterozygotes. I have a heterozygous black lab. So we're going to take a big B, little b, and we're going to make it to a chocolate lab. And here's where we start getting more interesting results. So I have a heterozygous black lab, and I'm going to cross it with a chocolate lab. So I got big B, little b, little b, little b. Well, I get one heterozygote, and I get another heterozygote, but look at these two here. These guys are all chocolates. These are all blacks. So when we think about this, genotypic ratio, we have 0 homozygous dominant. We have no big B, big B. We have 2 big B, little b's, and 2 little b, little b's. So this would be a 2 to 2 phenotypic ratio. 2 are black, 2 are chocolate. So we can see now that we actually generate slightly different outcomes. And if you look at it, anytime that you essentially take one homozygous dominant and cross it with a homozygous recessive, you always get heterozygotes, all of them. So when we take this, we had one heterozygote with a homozygous recessive. Well, we can imagine what happens when we change that up. Now, we have a lot of other kinds of things that we can do, and I'll do one more example where we can take this time, we have besides 2 heterozygotes. What happens here? So after that, we are pretty much seeing most of the outcomes that you can possibly see in standard genetics, or at least the kinds of problems you're going to work. 2 homozygotes, and we have 1 heterozygote to a homozygote. But let's do 2 heterozygotes. I get kind of bored of using the same colors. We'll use red. So let's do a big B, little b, a big B, little b, 2 black labs who happen to be heterozygous. So big B, little b, big B, little b, cross them, big B, big B, big B, little b, big B, little b, little b, little b. Now we have every possible combination. We happen to have a genotypic ratio that says, okay, I have 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. A 1-to-1 relationship. And if you were to see this in nature, and I got babies, and I looked at them, and they had a 1-to-1 ratio of their genotypic ratios, you could be safe to sure to bet that they were both heterozygous parents. So what does this look like? Well, we take those 2 and sum them, 3 to 1. There's the 1. So if I see a 3-to-1 ratio, I'm probably safe to bet that my parents are both heterozygotes based on the laws of probability. So that's how you do a basic one we call a monohybrid cross, in which we have 1 allele in common, where I make a Punnett square, and I look at these how to break down. Now, this is pretty simple. When we do dihybrid crosses, they can be a little bit more complex. And this will conclude my video at looking at the particular how to do a monohybrid cross.